Good morning and welcome everybody to the Grand Round lectures of the Pathological Society and the Shanks Pathology Foundation. This series has been running since 2021 and it's been very successful, covering a wide range of topics. Many of the lectures can be found on the PathSoc website. And so for those who'd like to revisit some of the lectures, they've been recorded for you to play back and listen to in greater detail. So the format of the lecture is that it's 45 minutes and we have question and answers at the very end. And we aim to finish promptly at 10 a.m. So today's lecture, it's an absolute great pleasure for me to introduce Professor Ming Du, who is the Professor of Oncological Pathology and a consultant molecular pathologist at Addenbrooke's Hospital, the University of Cambridge. And he's been there since August 2003. He obtained his medical degree in China, in Hebei, and he then moved in the early 1990s to London, to King's College Hospital, School of Medicine and Dentistry. And after a 3-year postdoctoral fellowship at the ICR, he joined Professor Peter Isaacson's research group. And then he was promoted to reader within that same department. So Ming has contributed significantly to our understanding of molecular pathology and the genetics of several lymphoma entities, including marginal zone lymphoma, and T follicular helper cell lymphoma, and also diffuse large cell lymphoma. The lecture he will be giving us today is the Doniac lecture, which he gave in Ghent in summer this year at the combined meeting in Belgium. So without further ado, it gives me absolute great pleasure to introduce Professor Ming Du, and I'm really looking forward to your lecture. Thank you very much, Ming.
Thank you very much, Sarah, for your kind introduction and all the kind words. And I'm really honored to give this lecture, as this is not only the recognition of my own research, but also my research groups and the vital contribution and by my student and the postdoc over the decade. And also, of course, my collaborators who has, helped me and contribute all aspects of the study. I'm really grateful to all my collaborators and former students and postdocs. And for me, it really has been very fortunate and to have the opportunity and to first work with Peter Isaacson at UCH, and then continue my work and with Andrew Wiley in Cambridge. And they are the giant in our society, and their acquisitive mind, particularly and probing the biologies and from the morphological observation under the microscope, and has been inspirational and for my research through my academic career. And so in this lecture, we'll very briefly and touch a few topics, because I think there are important questions I would like to share and with you. So this covers multiple lymphoma, diffuse large B, and nodal TFH, and lastly, and we'll touch the clonal lymphogenesis with and multi-malignant potential. So I was recruited to UCL by Professor Peter Isaacson and Lang Xingpan, and in 1993. So when I joined the department, there are two very exciting publications, and at that time, and they published two back-to-back papers in Lancet. And in the laboratory study, they demonstrated the gastric motor lymphoma proliferate in the response of heat inactivated H. pylori, and this response is dependent on the H. pylori specific T cells. And the second is a clinical study, and they demonstrated regression of primary low-grade and gastric and motor lymphoma following eradication H. pylori and by antibiotics. So in that initial study, they treated gastric motor lymphoma with antibiotics, and for 6 patients, and 5 of them they showed complete remission. With longer follow-up, the remaining case also showed complete remission. So this is really a seminal, exciting observation in the field, and we have to remember at that time we only start using the PCR and learning of the T helper cells, and also understanding the CD40 signaling between the B and T cells. So the discovery of this work is absolutely fantastic, phenomenal. Even let's say to these days, if I'm thinking we have a lot of omic technologies, whether we can achieve the same and seminal observation, it's really a big question. And when I joined the department together with my colleague at that time, and Dr. Pan, Dis, and Peng, so we have carried out several studies, and we mainly, and characterize dissemination pattern of multiple lymphoma and also the high-grade transformation. We also developing the antibodies and for BCL10 and demonstrate dissemination pattern. So in a subsequent study and in collaboration with Martin Dare and as the Institute of Cancer Research, and together we have cloned, breakpoint of the 114 translocation and identified BCL10 as an oncogene involved in this chromosome translocation. We also developing, as mentioned, and BCL10 antibody is showing stunning of BCL10 nuclear expression in the tumor cells. Actually, we can using this as a marker to trace disseminated tumor cells. And in the same year, and a study by the Judy Steerlam and from Belgium, they have uncloned breakpoint of 1118 translocation and identified the gene involved. And this, and cause a chimeric fusion between the API2 or BRCA2 to MALT1. And similarly later, and a study from the Bertold Strobel and from the Vienna, they identified together with a Spanish group, they identified another chromosome translocation in multiple lymphoma. This translocation has the same karyotype and like the BCL2 translocation, but nonetheless, this translocation involved in multiple lymphoma involved MALT1 rather than BCL10. So now we have 3 chromosome translocation in multiple lymphoma being identified. So we also developing the MALT1 antibody and examine the BCL10 MALT1 expression pattern in multiple lymphoma with different chromosome translocations. We also develop, an interface phase qRT-PCR method for the BRCA3 MALT1 translocation. With this method, these allow us to examine the translocation in multiple lymphoma different side. It's really to my surprise at that time. And the translocation occurs at variable incidence and in multiple lymphoma different size. For example, like the BRCA3 and MALT1 translocation and occurs in 25% of the gastric multiple lymphoma, 40% of the primary multiple lymphoma, but rarely detected in multiple lymphoma of other side. I was really puzzled by this, finding. And at that time, because, the gastric multiple lymphoma has been successfully treated by H. pylori eradication, which is been confirmed by several of the other independent studies. And in order to assess whether the individual patient's response to H. pylori eradication, we have to follow up the patients regularly and by gastric endoscopy and also gastric biopsy, normally let's say every 3 months, at least, for a year or 2, and to assess the response of the gastric multiple lymphoma to H. pylori eradication. So it's really where I, desired at time of the diagnosis, if we can predict which patients would respond to H. pylori eradication. So then we examined 1118 translocation and in the context of the H. pylori eradication therapy, and we have initially based on the frozen material And then in a subsequent study, we're using the formalin-fixed paraffin-embedded material. We have demonstrated the 1118 translocation is a reliable marker for all stage of gastric multiple lymphoma. We do not respond to H. pylori eradication. So therefore, in the clinical setting, then we can stratify the patients and for different management. And so if the translocation is negative, these patients can be, successfully treated and in around 70 to 80% of cases by H. pylori eradication using antibiotics alone. And if the translocation is positive, and this patient should be eradicated for H. pylori and also at the same time maybe considered for chemotherapy or radiation therapy, depending on the local expertise. So this is really, probably, let's say, the first time and demonstrate of the stratified patient management and in multiple lymphoma. And at that time, so if we're using today's buzzwords, so this would be the precision and the medicine. And the identification of BCL10 MOD1 through the lymphoma translocations also stimulate the functional studies of BCL10 and MOD1, and particularly from the immunology groups. So together, including study from us, we have demonstrated BCL10 and MOD1 link the antigen receptor signaling to the canonical NF-kappa B and the pathways. They are the positive and adapter molecules and forming the CARD11 and BCL10 MOD1 and the complex. And at the molecular level, and we know overexpressed BCL10 and MOD1, they can through CARD and interaction and generate of the oligomer, which can constitutively activate BCL10 and MOD1, and therefore then they can activate a canonical NF-kappa B and the signalings. And NF-kappa B activation is also, and transactivate of negative regulators like the TNF-alpha inducible 3, and also known as A20, and acts as a ubiquitin editing enzyme, which can and remove the ubiquitin genes, and also, let's say, sending these and targeted proteins into proteasome degradation. And for the 1118 chromosome translocation, which can also forming an oligomer through the heterotypic interaction between the Burr domain and the caspase-like domain. And again, then they can activate all the canonical NF-kappa B pathways. In addition, and the chimeric and fusion API2 MOD1 can also gain additional functions. In collaboration with Linda McAllister, and we have shown the API2 MOD1 chimeric fusion can interacting with, NIC, NF-kappa B inducible kinase, and by interaction with NIC, and can, through the C-terminal caspase-like domain, can mediate the cleavage of the NIC, therefore generate of the kinase stable C-terminal domain, which can activate it of the non-canonical NF-kappa B pathway. Similarly, and the API2 MOD1 can interact with LIMER and generate a oncogenic fragment called LIMO, which containing converts of the oncogenic and opportunity. So therefore, 1118 translocation and can activate both canonical and non-canonical NF-kappa B pathway. So at the time, let's say, at that time, so what are the genetic basis of translocation negative and multiple lymphoma? And we've been working on this and since then. And given multiple lymphoma deriving from the marginal B cells, there are several of the immune receptor signalings are very important for marginal B cell development. That's including the antigen receptor, Notch signaling, Toll-like receptor, and also BAFF and CD40 signalings, which activate all the non-canonical NF-kappa B pathway. So we have designed the experiment trying to, interrogate these signalings, identify the key players that may be targeted by somatic genetic change. And first, by by a re-CGH, then targeted next-generation sequencing. In our early studies by a re-CGH, we have a pinpoint and deletion at 6q, and in ocular adnexal multiple lymphoma, this deletion including homozygous deletion, which very much centers as the TNF-alpha IP3 or A20 locus, and so we reckoned, and this might be a tumor suppressor gene. Then we screening the mutation, then we found a high levels of the mutation of the A20 in ocular adnexal multiple lymphoma, and then we further examined and the A20 deletion mutation in multiple lymphoma different size. Then we identified, and it's rather frequent, much frequent than the chromosome translocation we have described earlier. In ocular adnexal multiple lymphoma, we also identified a significant association between the A20 inactivation mutation and also expression of the autoimmune and B-cell receptors and IgHV434, which is encoding and very often and auto antibodies, particularly called agglutinin. So, and in this scenario, an expression the autoreactive BCR and also inactivation of downstream NF-κB negative regulators, and the most likely functional synergistic and maintained constitutive NF-κB activation. So the identification of the A20 not only limited to multiple lymphoma, and in the later study, and found A20 inactivation mutation deletion is very common in other B and T cell lymphoma. So by NGS screening, we also identified a number of the interesting mutations in ocular in thyroid multiple lymphoma. We have identified and concurrent and frequent mutation involved TET2 and CD274, including the PDL1 and TNF alpha and the receptor SF14, which is encoding and the second and the B and T cell co-inhibitory and surface ligand. And so in thyroid multiple lymphoma, what we believe and genetic inactivation of these, inhibitory and surface receptors in T cells, and by genetic inactivation, TNF receptor SS14 and PDL1. So these will diminish of the inhibitory interaction and to the T cells, then the T cells becoming very happy, then providing and more exaggerated T cell help. In collaboration with Tirsa Marafiotti from UCL, and we have done the spatial analysis. Indeed, we can identify the activated T helper cells and in the vicinity of the B cell, and so it's further supporting and inactivating of these, co-inhibitory function molecules in the B cells that may have, indirect impact on the T cell helper functions. And in salivary gland multiple lymphoma, and we have identified recurrent mutation involved, G protein coupled receptor, particularly GPR34, which are accounting for, 16% of the cases. And very interestingly, and the GPR34 mutation are really specific to salivary gland multiple lymphoma. And these mutations are very much clustered and at C-terminal region, and most of them, they are deleterious and mutations, either generate stop codons or frame shift mutations, and which, and remove of the C-terminal phosphorylation domains, and therefore making the receptor on the surface more stable, and then they can deliver of the signalings to the G protein and intracellular signalings. As I mentioned, these mutations are rather specific and to salivary gland multiple lymphoma, and also the GPR34 also target by a translocation and involved, and X chromosome and also immunoglobulin and the heavy chain locus and upper regulate of the GPR34 and expression. So we have performed a series of the in vitro studies demonstrated these mutations are actually activating mutation, can activate of the GPR34 functions. We performed immunohistochemistry and demonstrated indeed GPR34 is highly expressed in the cases with, chromosome translocation or this truncated and the mutation. And in addition, in the cases without evidence of the GPR genetic change, we also demonstrate the strong of the GPR34 expression in 35% of cases. This indicate GPR34 and signaling is probably important for salivary gland motor lymphoma pathogenesis independent of the genetic change. In salivary gland motor lymphoma, we know the lymphoma, the monotypic, and neoplastic B cells always surrounding and the lymphoepithelial lesion. The lymphoepithelial lesion are preferentially involved duct epithelial cells rather than the acini epithelial cells. And so the emerge of this neoplastic lesion, a rounding of this lymphoepithelial lesion generate a halo appearance. So these are the pathological features for locating of these neoplastic B cells. As demonstrated here, it's showing a very nice and kappa and light chain restriction. And through the study, we have, a formulate of the a hypothesis. And so it's most likely this lymphoepithelial lesion is continually and generate, epithelial regeneration and also the damaged epithelial cells undergoing apoptosis. So when the duct epithelial cells, when they die, then they will release an enzyme called the phospholipase A1, which can convert of the phosphatidylserine on the apoptotic cell surface and generate the ligand we call lysophosphatidylserine. This can stimulate GPR34. So therefore, this lymphoepithelial lesion can generate a pyrochrine stimulation and causing constitutively stimulation of the GPR34. I think this is most likely explain why the GPR34 genetic change are specifically associated with salivary gland motor lymphoma is also and why the neoplastic B cells is always emerge around of the lymphoepithelial lesion. So it's nicely explained the pathogenesis of the lymphoepithelial lesion in salivary gland motor lymphoma. So similarly, we also observe low levels of the CCR6 mutation, which is not specific to any of motor lymphoma site. And this mutation, again, very similarly, and truncation mutations at C-terminal remove the C-terminal phosphorylation domain. And these, mutations are activating mutation and when you're performing the in vitro and functional analysis. Unlike GPR34, CCR6 appears highly expressed in most of the motor lymphoma. So the expression of CCR6 is independent CCR6 mutation. But nonetheless, with identification of the CCR6 mutation, it indicates CCR6 signaling is important for motor lymphoma pathogenesis. So in gastric motor lymphoma, we know, and it's most of them associated with H. pylori and chronic H. pylori infection. There are two of the metaplasia, and one is a pyloric metaplasia, the other is intestinal metaplasia. So we demonstrated of the high expression of the human beta defensin 2 in a subset of the cells undergoing of the pyloric metaplasia. And in addition, we know the human alpha defensin 5 is highly expressed and in the duct, let's say in the epithelial cells and surrounding of the stem cell population in the crypts. So the highly expression of these human defensin and beta defensin 2 and alpha defensin 5, I think on the one hand, then they can inhibit of H. pylori colonization and growth. On the other hand, then they can act as a ligand to stimulate the CCR6 and therefore promoting the B cell activation and clonal expansion. So now I switch gear to title of the diffuse large B-cell lymphoma. And I think the major clinical question here is diffuse large B-cell lymphoma is a heterogeneous entity, and around 40% of the cases, they do not respond to current R-CHOP therapies. Identification these 40% of cases not respond to R-CHOP therapy and devise them and for a different therapeutic therapeutic strategy is very, very important. It's challenging of the research field in diffuse large B-cell lymphoma. And so in the clinical setting, I think at the moment is we only performing phage analysis for MYC and BCL2 chromosome translocation in order to detect a double hit, and so we know these translocations occur around in 10%, and roughly and half of these MYC translocation positive case and has concurrent BCL2 translocation. They confer the double hit, and we know and clinically and they are more aggressive. And in our PML consortium, Precision Medicine on Aggressive and Lymphoma in the UK, and my colleague in Leeds, they have using the Burkitt's lymphoma signature and interrogate of diffuse large B-cell lymphoma, they have identified a subset of the diffuse large B-cell lymphoma showing very similar and expression signature to Burkitt's, and they call this subset as molecular high grade. This molecular high grade and diffuse large B-cell lymphoma, as we can understand, they're showing a very aggressive clinical outcome. But nonetheless, around 20% of the cases with MYC and BCL2 translocation, they do not show a molecular high-grade phenotype, and they are conventional germinal center B cell. And these cases are showing, you can see here, is much better and survival and very similar to conventional and germinal center B cell lymphoma. So the real question is why the double hit behavior and differently. Some of them behavior is very aggressive, the others is more like conventional GCB diffuse large B-cell lymphoma with a better prognosis. So in the same year, there is another study from the Lüneburg and the Biomarker Consortium. They have investigated a large series of MYC translocation diffuse large B-cell lymphoma, and they have identified and in the double hit cases, which MYC translocation associates with immunoglobulin gene, they're showing much worse prognosis than those double hit with MYC translocation involved non-immunoglobulin gene. So the question now is why MYC translocation partners matters in prognosis. And we have, and examined this and through, our consortium studies. And by mutation analysis, we have identified and frequently MYC mutation, particularly identify the hotspot mutation around the codon 57 to, 60. And this mutation is very much, located in surrounding of the first phosphorylation domain and in the codon 58. This phosphorylation is marked MYC protein for proteasome degradation, so therefore reduce of the MYC protein and half-lives. So there are this phosphorylation. So if you abolish of this phosphorylation site, and there are quite a few nice studies in the early literatures, actually you abolish this phosphorylation site, then you can enhance of the MYC protein half-life. So we in our cohort, and we demonstrated these hotspots mutation are significantly associated with molecular high-grade B-cell lymphoma, and also they are significantly associated with MYC translocation associated with immunoglobulin, either heavy chain or light chains. And also, if you perform immunohistochemistry, we also demonstrated the cases with MYC hotspots mutation, which abolish the first phosphorylation site. Then they show and homogeneous, very strong and MYC protein expression. And among the IgH MYC translocation, and we know probably around 6 being reported in literature, and through our ongoing study, we have identified more than 20 of MYC translocation partners. But BCL6 remained as the most common non-immunoglobulin gene partner of MYC translocation. So we performed a FISH to investigate of the MYC and the BCL6 infusion in in diffuse large B-cell lymphoma. We have demonstrated this is really quite high in the triple hit, and also, let's say, in the MYC and the BCL6 kind of double hit cases, we detect this 29% them, and due to the direct MYC and BCL6 fusion. If you look at the genetic subtype, those are the triple hit, and very, very similarly, and in terms of mutation profile, genetic subtype, and to the MYC and BCL2 double hit. On the other hand, when you look at the MYC and BCL6 double hit, and their mutation profile are very much different from the classic and triple hit of MYC and BCL2 double hit, and also as genomic subtype, and they are very much heterogeneous. So this is as a part of the evidence, and removing of the MYC BCL6 double hit and from the double hit entity in the WHO classification. And when we also examined the MYC protein expression, we demonstrated the cases with MYC IG translocation as expected, and they show very strong MYC protein expression. But in those with non-IG and always MYC and BCL6 translocation is only up to 50% of them, and demonstrate of the strong MYC expression. So in other words, the MYC protein expression very much depends on the MYC protein and partners. And we sequence a number of the cases of those with MYC translocation in diffuse large B-cell lymphoma. In the classic translocation with IgH MYC translocation, the breakpoint involved, in the switch region, very often the breakpoint in the MYC is upstream of 5' UTR or in the intron 1. So therefore, the super enhancer can driving MYC and protein expression. In some of the cases, it's also showing a classic promoter substitution. So therefore, MYC is under the transcriptional control of its partners. Again, this can cause a MYC protein expression. In non-IG MYC translocation, for example, with MYC and BCL6 translocation, these are the tail to head orientation, opposite orientation, and so in such genomic orientation, there is no apparent evidence this translocation can dysregulate MYC expression. So as expected in this case, and MYC protein expression is rather low. So to summarize of this part of the talk, and the high MYC protein expression in diffuse large B-cell lymphoma with MYC translocation depends on the genomic configuration that enables the MYC transactivation, and genomic configuration that very much depends on the super enhancer and promoter substitution. And the non-IG MYC translocation in diffuse large B-cell lymphoma is very much heterogeneous in partners and genomic configuration, may not always acquire a mechanism to constitute the transactivate MYC, expression. So high MYC expression in diffuse large B with MYC translocation. Oh, sorry. So now I switch the third part of the talk and focus on the nodal and follicular hyper cells. And as again, I have some important message and to share. And we all know TFH lymphoma and its diagnosis is very much difficult, and partially because very often we have core biopsies, and histologically because there are a lot of reactive background due to the EBV and the infection, EBV positive B cell triggered and immune response, and very often we have very low neoplastic contents. We do not have specific markers for neoplastic T cells, and we always using clonality as an auxiliary help for the diagnosis, but quite often it's not conclusive. And nonetheless, the patients with TFH lymphoma in general and are clinically aggressive, it's very difficult to distinguish from their mimics. And very often, and for a firm diagnosis of TFH, and in the past, quite often we require between 2 to 3 longitudinal biopsies to establish diagnosis. But now we understand and the genetics and alteration in TFH lymphoma, and we know and most of the cases arising from a background of clonal hematopoiesis and the type 2 DMT3A mutation are very common and in TFH lymphoma. So these occurred in the hematopoietic stem cell population. So therefore, this stem cell population can generate, their progenies carry these type 2 DMT3A mutations. If the progeny and, acquire a lymphoma-specific mutation like ROE, VAF1, PLC, gamma 1, etc., then they can, making these cells differentiate to TFH differentiation and gradually leading to malignant transformation. But nonetheless, these hematopoietic stem cells can also give rise to reactive B and T cells, which carry the type 2 and DMT3 mutation. So these also contribute to the pathology what we see in nodal TFH lymphoma. So that's why it's complicated of the diagnosis. So here is showing the heat map of these translocations, and you can see there are overlap mutations between type 2 DMT3 and ROE and IDH2. And for the ROE mutation, most of these mutations are localized codon 17, and majority of these mutations and leading to the glycine to valine and change. So therefore, you can using this mutation pattern and for an early detection. So we did this study. We generate a Q quantitative PCR for the ROE mutation. We retrospectively studied a series and with this ROE mutation with longitudinal and follow-up data. And so if this is point represent of the pathological diagnosis of the TFH lymphomas, and so we can detect the ROE mutation really in the early, biopsies, and sometimes quite a few years and before the diagnosis of the TFH lymphoma. So for this, biopsies, and because there is no evidence of the TFH lymphoma, and so therefore detecting this ROE mutation is this biopsy most likely represent a true reactive lesions. But nonetheless, in some of these cases, it's only a couple months ago and before the diagnosis, and so again we can detect in biopsies containing the ROE mutation. For this biopsy, most likely just because of the core biopsies or the lymph node biopsy not representative for the AITL. And so most likely a patient already at the time presenting a nodal TFH lymphoma, but and with have a sampling errors and unable to make a diagnosis. So therefore, detection of this ROE mutation is really useful for early detection of the AITL. And given, let's say, the hematopoietic stem cell mutation can generate multiple progenies, and the type 2 and mutation, DMT3 mutation, may skew the differentiation for TFH and T cells. So I think it's reasonable to speculate, and so this mutation may have the capacity to lead, multiple and malignant and, transformation. We have examined these properties, and by sequencing of the express TCR beta, then we can see here, and in each of these samples, a high proportion of these cases containing more than one, TCR beta functional rearrangement, and given their clonal size is different, so this is most likely represent independent and TCR beta rearrangement or indicate and different T cell clones. And on selected cases using the base scope in situ hybridization, and we have demonstrated and this type 2 mutation, the same type 2 mutation occurs also in these kind of minor and T cell clones, including these minor T cell clones, they are CD4 positive. So indicating there might be a potential for multiple parallel neoplastic and proliferation of T cells. And so now we have some of the case studies and demonstrated, and this is one of the case and study we have worked out with an AMR at Kigali and from Royal Marsden Hospital. We demonstrate multi-neoplastic potential, including metachronous and clonally unrelated nodal TFH lymphomas in clonal and hematopoiesis. So this is one of the patients we studied, and the patients, and this is and cervical lymph. This patient is 67 years and female, and I almost first received a cervical lymph nodes and a biopsy referral to her, and and this is showing a nodal monozome-like proliferation, but there are lots of the T cells that trigger AMR to perform next generation sequencing. We demonstrated this containing a BRAF mutation, which is activating mutation. It's also containing the TNF receptor SS14 mutation. Is clonal, but this is also containing a low level of the ROA mutation, and together with a high VAFs of the TAT2 DMT3 mutation. And the patient has a previous biopsy, and we request the previous biopsy and also reviewed, and this review and typical, TFH lymphoma angioimmunoblastic type. And we performed a TCR rearrangement analysis, identified two rearrangement, and they are very similar clonal size and most likely represent a, biallelic rearrangement. And we can detect the ROE mutation, but the ROE mutation, the VAF value is really very low, not, and less consistent with high tumor cell contents in that biopsy. And the patient has been treated and clinically follow up, and 26 months after initial diagnosis, and the patient showed a additional lymphadenopathy in soft tissue and cervical lymph nodes. We have the opportunity to study the cervical lymph nodes, and again, so here we demonstrate there are three TCR gamma rearrangement with different clonal size, therefore probably represent different T cell populations. We identify the VAF1 mutation, and again we still see a very low levels of the ROE mutation. So the morphological and observation indicate this is now a nodal TFH lymphoma NOS rather than angioimmunoblastic type. So as they are different, involve different T cell clones, and they deriving from the same background of the clonal hematopoiesis and with the identical TAT2 DMT3 mutation. So this elegant showing there is a divergent evolution from this, and clonal hematopoietic stem cell population generate two different TFH lymphoma in this case. In addition, it's also generate a nodal monozome lymphoma. And so to summarize this part, I think, let's see, these are genetic change in TFH lymphoma really generate a dynamic and, clonal evolution. They can leading to, TFH lymphoma proliferation, and this could be a minor clone to lymphoma. The minor clone could be detected and by sensitive ROA mutation or next generation sequencing. And also these mutant cell population can also, and giving rise to the progenies and T and B cells, and again this can manifest from minor clonal and to major clonal, sometimes forming a lymphoma-like lesion like cytotoxic T cell lymphoma or nodal monozome or follicular lymphoma. And lastly, I want to touch the clonal lymphogenesis with multi-lymphoma and potential. And so we always, from the lymphoma and diagnosis and from the lymphoma development, we quite often we spot, pre-malignant lesions in patients developing the first lymphoma. Then subsequently, after treatment, successful treatment, patient may developing a second lymphoma. So in the clinical setting, we often using the immunoglobulin gene clonality analysis to, investigate all the clonal evolution. But I want to emphasize this can demonstrate all the clonal evolution, but cannot telling us whether the relapse of the lymphoma, whether it's a linear progression from the first lymphoma, or they are, divergent evolution. Actually, they represent a new lymphoma. So I'm using two examples. The first example is the 1418 translocation, because this translocation occurred at pre-B cell stage during the VDG recombination. These translocations are really quite common, and particularly in the healthy adult population, and 50% of us carry these circulating and the translocation positive lymphocytes. So we have the opportunity to study this, and together with Dr. Andrew Wotherspoon from the Royal Marsden Hospital, and he has a very nice collection first and a series of the cases, and with solid cancer patients, and there is, a prevalence of in situ follicular B-cell neoplasia in the lymph nodes cleared in the solid cancer surgeries. And this patient has been, and follow-up and up to 16 years, and each of these patients developing a full-blown and follicular lymphoma. So we have analyzed all these cases. We demonstrate based on BCL2 immunohistochemistry in each of these, cleared lymph nodes at the time of the solid cancer surgeries. Then they can demonstrate extensive in situ follicular B-cell neoplasia. We sequence these cases together with the full-blown follicular lymphoma. We demonstrate the mutation and in this in situ follicular neoplasia in the pre-malignant lesions, and then you can draw the evolution map based on this mutation pattern and to predict the mutation accumulation. So in our study, then we can demonstrate together with the literature. Now we can demonstrate mutations and combination of this mutation in the in situ follicular neoplasia. We also demonstrated each time in the individual in situ follicular neoplasia, and then they can acquire additional mutation. So I think what's the scenario happens here when the translocation positive cell and they going through of this germinal center, like the germinal center reaction, then they can acquire the somatic hypermutation, and which accumulate and mutation in the BCL2 and also the other oncogenes. And so when they going through these B-cell follicles and accumulate enough combination of these mutations together with chronic B-cell signalings, and particularly acquisition and glycosylation sites in the BCR and also T-cell help, then they will gradually developing a full-blown and follicular lymphoma. And based on published data, we know and transformed follicular lymphoma, probably around 40% of them, they are not a linear progression from follicular lymphoma, but and 40% and deriving from this pre-malignant lesion from this in situ follicular neoplasia. And for those without follicular lymphoma components, like the de novo and double hit, and again they share very similar mutation patterns. I think it's very pertinent to speculate these cases also around 40% deriving from a pre-malignant neoplastic lesion which carry the 1418 translocation. And we also examined an early stage of follicular lymphoma. They are treated by local radiation therapies. So therefore, there is no systemic impact on the 1418 positive and cell population. And so for each of these cases, after local radiation, the patient is showing a complete regression. We also examined using our next generation sequencing panels. We demonstrate a linear progression of the relapsed follicular lymphoma or transformed follicular lymphoma, and in around 40% of cases. And in 2 of the cases, we demonstrate relapsed follicular lymphoma. Actually, they are different B-cell clones, and in another of the 30% of cases, we demonstrate they deriving from a common precursor, pre-malignant B-cell precursor subpopulation, because the pathogenic mutation predicted in the common lymphoma precursors are really very few, and by no means, and this indicate it's a full-blown malignant transformation. And so again, further, two cases we demonstrated it's kind of, let's say, divergent progression from the early and malignant clones. So if in follicular lymphoma, I guess we can also adding in the recurrent or relapsed follicular lymphoma, probably around 40% of them. They are not a linear progression from the, first diagnosed lymphoma, but they are really deriving from the clonally related and BCL2 translocation positive pre-malignant lesions, or from most likely this in situ follicular neoplasia. And again, we know this 14-18 translocation can also giving rise to multiple lymphoma and also giving rise to a number of the different entities, low-grade B-cell lymphoma, even histiocytic sarcoma or lung cancer sarcoma. So lastly, I want to using the thyroid motor lymphoma model to demonstrate all the same phenomena. In thyroid motor lymphoma model, and we know is B-cell lymphomas deriving from background of the Hashimoto thyroiditis, which is autoimmune disease, and very often we will detect clonal and B-cell population in the Hashimoto and the thyroiditis. Again, and we have studied a small cohort of cases with multiple biopsies, and again we demonstrate and and very small numbers in linear progression, but in majority of cases, there are really divergent evolution of these two different lymphoma from the common lymphoma precursor cell population. And so to conclude this part, I think the current diagnostic investigation of the metachronous or synchronous lymphoma and in follicular lymphoma or chronic and LPD background is not optimal, because analysis of the IgH BCL2 rearranged immunoglobulin gene provide of the evidence for clonal relationship, but they are unable to reveal their evolutionary history. So therefore, it's unable to illustrate whether they are new lymphoma or truly, lymphoma relapse from the initial lymphoma diagnosis. And so mutation profile, I think at the moment is the alternative and to illustrate of this evolution path. So in terms of the clinical management, I think it is also important message. So if it's true lymphoma relapse, so this is probably evidence of treatment failure. If it's new lymphoma, I guess, and they share the same biology, so they may remain sensitive and to the same standard and first-line therapy. So in terms of biopsy relapse lesions, even without, suspect high-grade transformation. And so again, in the current practice, I think we only do a biopsy if we suspicious high-grade transformation. In light of this finding, do we need to do rebiopsy any of these relapsed lymphoma lesions? And recently, and I wrote a review and for blood. I'm really very pleased, and they using my lymphoepithelial lesion and as the face cover for that issue. And so in that review and in my concluding remark, I wrote the pathological process and histological presentation remains as a timeless cornerstone for mechanistic investigation. I think this is still, very much appropriate and let's say conclusion and for my talk. And I thank you so much for your attention. And lastly, acknowledge my mentor Peter Isaacson and also my and collaborators, my student postdocs and collaborators and all over the world, and also the funding bodies and including Pathological Society, and which have been really very generous and to support me and and they support many of my international collaborations and as well as and studentship and the small equipment grants. I'm really grateful to all the support from the society and also the other funding. Thank you so much. Thank you
very much, Ming. That was fantastic. A real tour de force. You showed phenomenal advances which have in our understandings of lymphoma over the last 2 to 3 decades. The very first WHO lymphoma classification. And you've contributed significantly to all of the understanding of just those 4 or 5 entities that you outlined there in your talk. And so all of these contributions have fed into the latest WHO lymphoma classification. So thank you extremely very much. I have a question, but I see that there's a question in the QA box, and I just encourage the people who are online to also write their questions. So there's a question from James Morris, the concept that H. pylori has a pathogenetic role in gastric adenocarcinoma lymphoma was a paradigm change in pathology. Is there any evidence that an intratumor microbiota has a role in salivary, thyroid, or lymph node lymphomas?
It's a very good question. And so far, particularly for salivary gland, it has been, investigated, including us. But so far, there is no firm evidence indicate there is other microbiome involved in salivary gland motor lymphoma. But for both salivary gland and thyroid motor lymphoma, as we know, and they are very much developing from background of the autoimmune and disease. And I think in these conditions, and autoimmunity are really the driving force for developing lymphoma in salivary gland and thyroid and lymphoma, because these, driving force not only for developing of motor lymphoma at this site, I think they are, more generically important for developing all B-cell lymphoma, including, rarely follicular lymphoma and also, diffuse large B-cell lymphoma. Among motor lymphoma other sides, I think, and although it's kind of anecdotal, but the evidence from Italy is firmly confirmed, and committee CTI is involved and in pathogenesis ocular adnexal motor lymphoma, particularly and in certain geographic region, and that is involved. And again, there is evidence eradication of the committee CTI and lead to partial or complete response of ocular adnexal motor lymphoma.
Thank you. I can't see any further questions. Excuse me. I'm going to ask you a question if you don't mind, and I think we've still got time. It's very interesting with the multiple lymphomas that there are the different translocations at different sites, and yet these translocations still lead to the same morphological changes. You just showed your review at the very end and the importance of the lymphoepithelial lesions, and so even though these translocations occur, they don't result in differing morphology within the multiple lymphomas. Do you have any explanation for that?
Not totally. And I think in salivary gland motor lymphoma, we have that evidence. And I think that explains why GPR34 specifically associated, because the neoplastic B cells require paracrine stimulation. That's paracrine stimulation, the ligand, and can only be generated and by, salivary gland, because PLA1 is expressed in salivary gland, not expressed in other, and the GI gland. And I think that's provide a good explanation for thyroid lymphoma. What I really believe, because most of these are deriving from Hashimoto thyroiditis, and the BCR is autoreactive. We know the BCR encoding has autoreactive properties into thyroid globulin or peroxidase. So these are very much, organ and site specific. And so I think that's explained in thyroid motor lymphoma. Lymphoma cells quite often and they return in the thyroid. And also in terms of lymphoepithelial lesion, thyroid lymphoma, they have very profound, and the lymphoepithelial lesion is they also, forming a different lymphoepithelial lesion like the thyroid balls, because the lymphocyte enter to thyroid follicles and proliferation within these thyroid follicles forming a thyroid ball. I think the case, such a phenomenon is due to, because the BCR is thyroid globulin or peroxidase and specific, and they want to, home into, area with a lot of antigens. But for other, translocations of phenomena and like gastric motor lymphoma, I think we have some explanation like and human defense in the innate immune response, and these could be potentially explained. But I really can't understand why the BCL10 MOD1 translocation are really preferentially linked to gastric and pulmonary motor lymphoma, particularly given they are, sit downstream of BCL signalings, and we recognize BCL signaling is important for all lymphomas, but why, let's say, they are specifically associated with gastric and pulmonary motor lymphoma? It's still a puzzle to me.
No, that's great. A very quick question. The multiple lymphomas do tend to have this homing phenomenon, and also that they sort of can, you can have them occurring in one site and then relapsing in another extranodal site, years later, but still showing the same B cell clone from which they've arisen. So there's almost like a senescence going on, or dormancy. And I don't know, do you have any thoughts on that?
Yeah, very, very intriguing question here. Yeah. So, and I think, let's say, we have to imagine, because the B cell population as the mucosal site, they respond to the local immune reactions. So therefore, they acquired as the mucosal site, specific site. So the etiology is very much different, and the homing receptor expression is also different. So when they, let's say, engaging of the immune response at different sites, I think they are induced to express their site-specific homing receptors.
Right.
And I think that on the one hand, that would determine their homing properties. On the other hand, because sometimes these BCR signalings and the B cell autoimmunity, they may have cross reactions. So for example, let's say gastric motor lymphoma, sometimes they can present as a salivary gland. So the reason for that, and I think we partially understand, because the BCR could be, encoding autoimmunity like rheumatoid factors. So therefore, then they can, and recognize similar antigens in the salivary gland, and they homing to both, gastric as well as the salivary gland side. So that's my speculation.
Okay. I think we unfortunately have to leave it there. I might continue the conversation with you afterwards. And I'd also say to everybody that Ming has offered, if you've got any burning questions or comments, please contact him directly via his email. So thank you very much, Ming, again, for your fantastic lecture. I'd also like to express my our gratefulness to the Shanks Pathology Foundation for co-sponsoring these round lectures on a regular basis each month. And with that, I'd like to thank you for tuning in and participating. And we'll have an outgoing slide which will list the upcoming round lectures for the end of October, November, and also December. So, and if you scan the QR code, you'll find more information on our Pathological Society website. So thank you very much for your attention, and we'll see you next time. Thank you. Goodbye.
Okay, thank you.